The T cell receptor loci comprise sets of gene segments and are rearranged by the same enzymes as the immunoglobulin loci. Like immunoglobulin heavy and light chains, T cell receptor alpha and beta chains each consist of a variable V amino terminal region and a constant C region. The organization of the gene segments is broadly homologous to that of the immunoglobulin gene segments. The TCR alpha locus, like those for the immunoglobulin light chains, contains V and J gene segments, V alpha and J alpha. The TCR beta locus, like that for the immunoglobulin heavy chain, contains D gene segments in addition to V beta and J beta gene segments. The T cell receptor gene segments rearrange during T cell development to form complete V domain exons. T cell receptor gene rearrangement takes place in the thymus. Essentially, however, the mechanics of gene rearrangement are similar for B and T cells. The T cell receptor gene segments are flanked by heptamer and nonamer recombination signal sequences, RSSs, that are homologous to those flanking immunoglobulin gene segments and are recognized by the same enzymes. All known defects in genes that control VDJ recombination affect T cells and B cells equally, and animals with these genetic defects lack functional lymphocytes altogether. A further shared feature of immunoglobulin and T cell receptor gene rearrangement is the presence of P and N nucleotides in the junctions between the V, D, and J gene segments of the rearranged TCR beta gene. In T cells, P and N nucleotides are also added between the V and J gene segments of all rearranged TCR alpha genes, whereas only about half the VJ joints in the immunoglobulin light chain genes are modified by N nucleotide addition, and these are often left without any P nucleotides as well. The main differences between the immunoglobulin genes and those encoding T cell receptors reflect the fact that all the effector functions of B cells depend upon secreted antibodies whose different heavy chain C region isotypes trigger distinct effector mechanisms. The effector functions of T cells, in contrast, depend upon cell-to-cell contact and are not mediated directly by the T cell receptor, which serves only for antigen recognition. Thus, the C regions of the TCR alpha and TCR beta loci are much simpler than those of the immunoglobulin heavy chain locus. There is only one C alpha gene, and although there are two C beta genes, they are very closely homologous, and there is no known functional distinction between their products. The T cell receptor C region genes encode only transmembrane polypeptides. And that's the T cell receptor loci comprise sets of gene segments and are rearranged by the same enzymes as the immunoglobulin loci. Have a great day.